This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 250, recorded on September 2nd, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Michigan, Michele Swanson.
Hello. Nice to see you both.
We are not recording video, but Michele is out in the woods, which is really nice.
And I'll share that I'm elated because my daughter gave birth to a healthy baby boy this morning.
Do we have a name?
Yes, it's Paul Carl.
Cool.
That's nice.
Yeah.
So is that your first grandchild?
It is. And I haven't yet talked to the happy family. I mean, she's probably still,
Where does she live? What part of the country?
D.C.
D.C.? Excellent.
Yeah.
Congrats. Very good.
So what are you going to be called? Grandma? Granny?
Grammy.
Grammy.
As in Grammy Awards. All right.
Yeah.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. Today we have the remarkable temperature of 82 Fahrenheit and 28 C.
You didn't have this hurricane that we had, did you?
No, we're far enough east that it just unfortunately clobbered the folks in Louisiana.
So it went inland past you and up towards us, right?
Yes.
Yep.
We got just flooded last night. A mess. It's a mess. Commuter trains are still not running.
It's amazing that you made it into the city, Vincent.
Yeah, I had to change the liquid nitrogen, unfortunately. Otherwise, I might not have come in, but it's okay. And our basement got flooded this morning, so I was vacuuming water. It's lovely.
Oh, boy.
It's okay. Worst things can happen. All right. We have a couple of papers for you today for your listening pleasure, for your education. And let's start with a snippet by Michael.
All right. This one is entitled An Integrated Hypochlorous Acid Producing E-Scaffold is Active Against Monomicrobial and Polymicrobial Biofilms. And it's from a group at the Mayo Clinic, the Division of Clinical Microbiology and Division of Infectious Diseases, as well as the Jean and Lyndon Volin School of Chemical Engineering and Bioengineering at Washington State University in Pullman, Washington. And the authors are Florin, Ravel, Mohammed, Greenwood, Quaintance, Cano, and Banal. And the senior author is Robin Patel, one of ASM's immediate past presidents. So I've been following this literature for a while as I find this topic of in situ generation of strong oxidants to control infections to be most fascinating, for it offers us an opportunity to think of strategies to develop alternatives to antibiotics that closely mimic what our immune system does to control infections. Longtime listeners might recall that we discussed an earlier paper by Sultana, Call, and Banal in 2017 when we discussed their work entitled Eradication of Pseudomonas aeruginosa biofilms and persister cells using an electrochemical or the E-Scaffold, an enhanced antibiotic susceptibility that was published in the Nature Partner Journal series, Biofilms and Microbiomes, that was published in November of 2016. And we covered it on TWiM 143 in 2017. And we'll put a link to the show note for that TWiM episode. Today's paper, again, comes from an engineering lab and a very clinically focused group at the Mayo Clinic. And their hope is to advance the results from this study to ideally develop an electrochemical bandage to treat wound infections. First, some background. Hypochlorous acid is generally considered to be a highly destructive, non-selective oxidant that has strong bactericidal activity. And what many don't appreciate, that hypochlorous acid has a significantly higher oxidizing potential, meaning it's better than hydrogen peroxide, with its electrochemical potential being 1.63 for hypochlorous acid, while for peroxide, it's only 0.8 when you consider these one electron reactions in protic media. For those of us...
Michael, hydrogen peroxide will bleach your hair. Will hypochlorous acid also bleach your hair?
Oh, yeah.
Really?
Oh, yeah. It's bleach.
Okay.
And for those of us who have forgotten our chemistry, a protic medium translates into a hydrogen atom bound to an oxygen, as in the case of a hydroxyl group, a nitrogen as an amine group, or a fluoride as in the case of hydrogen fluoride. And the molecules of protic solvents readily donate their protons to solutes, often via hydrogen bonding, with water being the most common protic solvent. So you knew what a protic solution was, because we all know that the chemistry of life is water. The other variable to appreciate is hypochlorous acid is electrically neutral. It enters microbes by passive diffusion. And so once inside the bacterial cell, it alters the energy transport system of the cell, leading to rapid ATP hydrolysis, which ultimately ends up in them dying. So now let's consider the infectious agents that the investigators evaluated. Well, here they are looking at microbes that cause infections that are clinically remarkable to them. So these are principally clinical isolates that they are looking at. And the other significant component is that these microbes happen to form biofilms. And recall that biofilms are often more challenging to treat than those infections caused by the planktonic forms of the microbes, for a variety of mechanistic reasons. The first being the phase of growth that we commonly use to treat the majority of infections depends upon the microbes growing. And the majority of microbes in biofilms are in a stationary phase of growth. And so consequently, the antimicrobials are just ignored by the microbe. And then there's also more complex reasons why these infections are challenging to treat. And the first reason is when microbes are growing as biofilms, they produce something called extracellular polymeric substances. These are nothing more than biopolymers of microbial origin. And often when microbiologists introduce this to a lay audience, they introduce it with the vernacular term, you think it's snot, but it's not. And it's a very gooey, viscous, and in fact, the oil drilling industry uses a lot of this EPS material for oil drilling mud, because it's cuts down on the friction associated with the drill bits. But here, EPS actually changes the ability of antibiotics to penetrate the biofilm. It often changes the absorption content of the film, the water content, the porosity, density, hydrophobicity, and most importantly, mechanical stability. They actually stick there. So what all this boils down to is biofilms effectively then develop an overall tolerance to our innate immune defenses. They sort of just sit there and persist. So if you get a biofilm-based infection, like happens in chronic things like wound infections that often manifest horribly in diabetic feet, where a diabetic bumps their foot and they develop an ulcer, it can be quite horrific. And so it's really very challenging to treat those infections.
So, Michael, before you leave the snot image analogy, you mentioned that antibiotics have a difficult time getting through, but will these hydrochloric molecules more easily?
Yes.
So that's part of the brilliance of this strategy.
It's part of the
biofilms are local, so you could put a bandage right over it. It's not like you're treating something that's in the bloodstream. Yeah.
And since this paper was published in March. And I had no trouble accessing it offline. You can take a look at their figure, and they have a beautiful first figure that shows what their device would look like. It almost looks like a wristwatch that you could imagine being strapped over the wound to effectively treat that. And we'll come back to that in a second. Another troubling property of biofilms, besides this tolerance to antibiotics because of their growth phase and all the other mechanical things, is that if you have a mixed biofilm infection, which is often the case, you can actually share information amongst the microbes, so you can acquire antibiotic resistance, either associated with mutations or horizontal gene transfer, which can then spread through the biofilm and make it even more refractory should we have an antibiotic that can go in and treat. So as Michele pointed out, the premise of the work here is that we desperately need alternatives to antibiotic therapy, specifically to treat these wound-based biofilm type of infections. Now, Vincent asked, well, what about bleach? Well, topical agents like sodium hypochlorite, which we also know as common household bleach, or many of us have gone and had our blood drawn for medical tests or have donated blood. You've been your skin has been disinfected with povidone iodine. They can often be used to lead to biofilm eradication. The problem with these agents is that when you set off a molecular grenade, and that's what the strong oxidants are, molecular grenades, is the resulting cytotoxicity, just like when it takes the color out of your hair, the resulting cytotoxicity to the host may impair healing. So the story here is the next evolution of their quest to develop an electrochemical bandage to treat these wound infections. The critical element here being the sustained, and that's the underscore here, sustained delivery of hypochlorous acid, because in their first experiment, they perform a Goldilocks experiment. They want to effectively determine the right concentration of the hypochlorous acid that can be evolved by their device while not effectively harming the host. So in their first figure, the team describes how they plan on generating this continuous antimicrobial agent, the hypochlorous acid, that is not generated at too high of a dose that becomes toxic to the surrounding tissue. The design or the chemistry is illustrated in their first figure, specifically in panel B. And where they get the chlorine donor is from the normal sodium chloride that is effectively leaching out from the wound exudate. And so that serves as the chlorine donor that will generate chlorine gas, if you will, or chlorine, that is generated by passing the electrical current through the device. And then the hypochlorous acid is produced electrochemically by oxidizing chloride ions to the chlorine in solution, that then the chlorine spontaneously dissociates in the water matrix to produce the hypochlorous acid. And then
that is just such an elegant strategy. The fact that you've got sodium chloride salt in the tissue, so you'll have a limitless supply of one of the substrates for this reaction, right?
Yes. And it's also the most clever bit is it's rate limiting, because we all know that the regulation of sodium ions in our tissue fluids is tightly controlled. And so you're not going to have too much salt that will then generate too much acid. And they go through great pains talking about the amount of electrons passing through their working electrode. And they show us how they calculate this. And the way they do this is with a really elegant piece of equipment called a Potentiostat, which, as its name implies, measures the electrical potential in the surrounding area. So the their current data, and that's current as electrons are flowing, not the time. Current data were integrated over the time to calculate the total number, if you will, electrons or charge delivered through the working electrode. And so the hypochlorous acid concentration was then estimated by just simply doing what we all learned how to do in freshman chem, stoichiometry. So 1 mole of hypochlorous acid will result from the 2 moles of electrons being donated from their device. And it's really very elegant. So we have a continuous current generating a continuous low dose concentration of hypochlorous acid aimed at targeting biofilms without exceeding the concentration that is toxic to the host tissue. Now, the device they referred to as an integrated electronic scaffold, or E-Scaffold, is an ingenious device, and it consists of 3 electrodes. And if we all remember how we measure pH, you always have a working electrode and a reference electrode, but this one has a 3rd electrode, which is a counter electrode. They're polarized at 11.5 volts, and they're measured against a silver chloride electrode like we were taught in our analytic chem course. So,
for those of us who took analytic chem,
once upon a time.
Not all our listeners or podcasters did.
Well, what's really neat about this paper is they go into great detail in the methods on how it was done. And if you have a rudimentary understanding of stoichiometry, what most people listening to this show likely do, you'll get a sense of how they're able to figure out what's going on. So today's work describes an improvement to their design where they simply integrated the reference electrode into the E-Scaffold. So what they're doing is they're miniaturizing the device, and they're really trying to adapt it for in vivo use. For example, on an infected wound, they're miniaturizing it. So they first asked whether their design would control clinically relevant microbes growing as a biofilm. And as I said, it was a Goldilocks experiment. They took 33 isolates of microbes that were isolated from various sites, various infected sites from people. And, the lab at Mayo has this wonderful collection of isolates, and it's listed in one of the tables. And then they took 12 combinations of dual species biofilms, and they simply measured the reduction in viable cells contained within the biofilm at 1, 2, and 4 hours, simply by turning on the current. And so what we're doing is we're moving from the bench to almost the bedside in this set of experiments. The objectives of their study were really very straightforward: improve the design of the former electronic scaffold to include this integrated reference electrode, and then test its spectrum of antibiotic activity, including this polymicrobial biofilm scenario, using clinically remarkable isolates. And they did this by principally measuring the current produced after 1, 2, and 4 hours of polarization, turning on the juice, if you will, where they found that the current flux generated 1.4 or almost 1.5 milliamps at 1 hour, 1.26 milliamps at 2 hours, and then 1.3 milliamps at 4 hours, which they then convert into cumulative concentration of acid produced, which turned out to be 7.89 millimolar at 1 hour, 13.46 millimolar at 2 hours, and 29.5 millimolars after 4 hours.
And the reason that was important is, as you pointed out, this can be quite toxic to the host tissue if it gets too high. So they're working in a really low subtoxic concentration area, which is great.
But most importantly, 29.5 millimolars is the theoretical maximum based on the concentration of the sodium chloride available from the affiliated wound. So it's really looking at the whole elephant, if you will. So the data are clear and remarkable. They show us beautiful color figures showing that, yep, the bacteria are being killed. The starting concentration of viable cells was 7.8 logs, which is 63 million cells for those of you who don't speak log. And they found that after 4 hours, they had 6 logs of killing, or at the end of that period, they had 97.8% reduction for, and this is the average for all of the strains considered. Now for the fascinating microbiology bits. And this is what really excited me. There was variability in the time frame of effect, with differences noted between aerobic and facultative anaerobically gram-positive and gram-negative bacteria. So, for example, viable cell measurements for all the gram-negative bacteria were reduced below the limit of detection by 4 hours. So this device worked really well with gram-negatives. But for the aerobic and facultative anaerobe gram-positives, a reduction to below the limit of detection at 4 hours was only observed for one strain of Staph aureus, and Enterococcus, and a Strep mutans. So why the differences between the type of bacteria? Now, here they offer us a hypothesis to consider, one that offers us, which now that many of us are starting to teach micro to our new class of students, is that we always tell them gram-positives have very thick walls and gram-negatives have very thin walls. And remember, the hypochlorous acid must pass through the cell envelope in order to exhibit the antimicrobial activity. And so first you got to get through the wall in order to get inside the cell. And because the hypochlorous acid is electrically neutral, it's entering by passive diffusion, and the gram-positives indeed have a thicker cell wall, between 20 and 80 nanometers on average, depending on the species that you're looking for. And they looked at a whole bunch of different gram-positives. Gram-negatives typically have a bimolecular layer of peptidoglycan, and it's only about 10 nanometers in thickness. So therefore, passive diffusion through gram-negatives, it shouldn't be surprising, is going to be quicker than through gram-positives, so you would expect the gram-negatives would die faster. And they offer that these differences between gram-positive and negative cells are going to be examined further in their future work, where they're going to focus on refining the mechanism of action of hypochlorous acid beyond just telling us it messes up with energy generation. Now, the last experiment, it's rare to find a monoculture biofilm. So they then experimented with 12 polymicrobial biofilms that were clinically remarkable. That is, they just didn't simply mix bacteria together. They mixed relevant bacteria that often show up together in these infections.
But just pairwise, right?
Yes, there was only two. There was only two. They didn't
So again, probably not really mimicking what happens in nature, but certainly a step in that direction.
But they had 33 strains. They would be still working if they put it all together. So based on their very promising results, next steps will be to advance this device to an electrochemical bandage to then test it in vivo to treat wound infections. So what really struck me is I think the next experiments will likely involve playing with the current so that you could, or maybe supplementing it with some additional sodium chloride to effectively get rid of those gram-positive infections but the trick is it's still got to be the Goldilocks. You can't have too much hypochlorous acid that would impact the vitality of the host surrounding tissue. So that's basically it. E-scaffolding is continuing, and we may have a bandage, hopefully, in the near future.
Michael, what would they have to do to get this into people?
My guess is they'd have to demonstrate utility. They'd probably use some of these rat models in which they put biofilm infections in joint repair established. And there are some good animal models out there that look at biofilm infections
Or
in
like
rats.
A staph abscess.
Yes.
MRSA abscess.
I see. So animals first, and then if shown to be safe and effective, then
then it will go to people.
Trials. What is your feeling about the likelihood of this working, Michael? You have a sense?
I think anything that offers continuous delivery will offer you a great opportunity to effectively go after these viable but non-culturable cells. Because you could imagine that you could turn the current on, turn the current off, and it would effectively allow the persister cells or the VBNCs to come out of the woodwork, restart metabolism, eating the detritus of their dead cousins in the biofilm matrix, and then turn the current back on. And so the acid would then destroy them. And we know that VBNCs have a really rough time dealing with these strong oxidants because they affect, there's always some basal level metabolism. And because the hypochlorous acid passively diffuses in, you don't need an active process. It's just going on in. And the beauty of the acid is because the biofilms are hydrated, it's moving with the protic solution, namely the water or the sodium chloride that's in solution. So when you turn on the juice, the electrons can move freely because they're the size of an electron. They're not the size of a polymer or an antimicrobial, and they just move through the system.
So to get back to Vincent's question, I was just trying to think of different types of infections we see in humans. Certainly like a diabetic foot wound would be an opportunity. Maybe the next level of complexity would be a tooth abscess. But then like endocarditis and growths on heart valves might be a trickier scenario.
Well, we said that 30 years ago before angioplasty replaced coronary artery bypass surgery.
Yeah, or what about, could you install these on, prosthetic valves? Because they also get colonized, don't they?
You could make little tiny, you could put it on a stent. And I'm sure Johnson & Johnson, the great manufacturer of all things stents, has probably looked at E-Scaffolds. And you never know what invention disclosures have been filed. Because it's the miniaturization. Remember, bacteria are very small. and you only need a little bit of current in situ. And the beauty of this is they're going from something very big in the lab and they're miniaturizing it. And, most of us have discovered the remarkable Apple Watch with all of its smart sensors. I mean, did you ever think it could do pulse ox just by sitting on your wrist and, do an EKG that's accurate? And I mean, sensors are getting pretty sophisticated. So I'm hopeful that it's a unique group because we have, this group from Pullman, Washington, engineers, and then we have the premier micro groups at Mayo Clinic working out the microbiology and really solving the puzzle of how to address the biofilm-based infection. So
biomedical engineers.
Yes.
We need more of those. Yeah, cool.
For sure.
It's really elegant.
Yeah, I think it's really very cool. And obviously you need this kind of collaboration to do this.
Yeah.
So, because a microbiologist would never know how to make such an instrument.
Well,
miniaturize it.
Well, that's why I went through some of the crazy chemistry, and it's really a nice paper because it explains chemistry that you can actually see how it works. You get it.
Yep. It's nice. Very good. Thank you, Michael. Well, I have a couple of papers also about technology, a little less sophisticated, but nonetheless useful, I think. And these will answer the question, are two snippets equal to a paper? because they are both really snippet size yet. So I have two, and they both deal with the same approach. The first one is from Applied in Environmental Microbiology, and it's called A Novel Method for Sampling and Long-Term Monitoring of Microbes that Uses Stickers of Plain Paper.
It's awesome. And I love that they have got that in the title.
Isn't that great?
Yeah.
Bobel, Witte, Mester, Pfister, Schroeder, and Rossmanith from the University of Veterinary Medicine in Vienna, Austria. And both of these are all about trying to sample surfaces for, in this paper, bacterial pathogens. In the other, it's going to be for viruses. And the focus of this paper happens to be Listeria monocytogenes, which we have talked about before, a pathogen that's often in foods. And so people take great pains to make sure it is not in food processing environments. There are standards to deal with getting rid of it, cleaning, disinfecting, monitoring surfaces and foods. But, we're always trying to improve our detection, right? Because you can imagine that if we're below the limits of detection of our, whatever the sampling assay is, we're going to miss it.
Until someone gets sick.
Until someone gets sick, yeah, and then we have a problem.
But also to make it accessible to broad audiences.
Yeah.
So that it's not incredibly expensive.
Now, as you might imagine, one way of looking for these microbes is to try and culture them, right? But as you must know, if you've listened to TWiM, that doesn't always work. It's not the most sensitive. So we need some alternative methods. Furthermore, not just culturing, but usually we swab a surface to see if it's contaminated. You take a swab, you rub it so many times on a surface, you put it in a tube, and you bring it to the lab.
Like a Q-tip.
Like a Q-tip.
Yeah.
Of course, that's a snapshot, right? And you don't know what happened yesterday. So if they cleaned the surface yesterday and you sample it today, that's not an accurate indication of what went on.
And the other thing to remember is that if you cleaned your surface, there may be residual cleaner on the surface that will then inhibit the culturing or may interfere with the nucleic acid amplification. So oftentimes you dilute out, taking into consideration that the cleaning solvents that are used routinely may interfere with PCR as well as culture.
That's right. And also, of course, the nature of the surface makes it challenging, like a door handle, a light switch, not as easy to get a good sample of, right? So the recovery can be very low. Now, in recent years, PCR, polymerase chain reaction, has been explored, which is very sensitive and gives you faster results. But of course, it does not distinguish between living cells and dead cells. So you don't know how long the contamination has existed or whether there are actually living bacteria present. Nevertheless, if you can combine it with other methods, it may be useful. So what they do in this paper, in both papers actually, is the idea of sticking something on the surface that can remain there for a while. So you get a longer picture of contamination, not just a snapshot. Whatever you put on the surface has to have, of course, certain properties. It's got to be more or less resistant to moisture, to abrasion, and it should not inhibit PCR and so forth. And what they decided to do was use plain, sterile paper. They stick it on surfaces, and they do some spiking experiments, and they recover the bacteria and compare culturing versus PCR methods to recover it. And, I just think of a sticky notepad. It's a plain piece of paper, right? You just stick it on the surface. You leave it there for a while. You can even have it get cleaned. It can be small enough so that the cleaner just cleans over it. And they actually looked at this here as well. So it can stay in place for a long time. So think of it as a sticker.
Literally a sticker.
A sticker, the kind that you would write a note on, and a Post-it note, right? That's one of the things that they're called. But I think they're sticking it on the whole back. A Post-it note typically has the adhesive just at one edge, but these are really stuck on the surface. So they have these text marking stickers, and they do some experiments to figure out if that would work. So they have Listeria, they have a mutant strain of Listeria, and they have an E. coli. They put it on stickers in different concentrations, and then they wait a day, 24 hours, and then they either do plate counts or try to enrich by growing whatever was on the paper in broth for microbiological analysis. And when they do that, they basically get random detection of E. coli at any concentration. But they need, for Listeria, they can only detect down to 100 colony forming units. And basically, they don't like the idea of doing culture off of the paper. It's not very sensitive, and it's not really a good way to do it. However, when they do PCR, so you take the paper and you extract it and do PCR on it, they get better, they get more quantitative results at different concentrations of bacteria. So they decided to focus on PCR. So putting the paper sticker on a surface and then assaying what's on it by PCR. So they do an experiment to ask how long does this signal last on the sticker. So they take 6 different stickers. They contaminate them with different amounts of Listeria, 500 colony forming units. They leave them there 14 days. And then they tried to plate they extract them and plate them out on plates, agar plates, or extract the DNA directly. When they plate them, they only get about 30% recovery and not much decrease after 14 days of storage. So that's a good sign. How about cleaning? So if you leave these stickers on food processing surfaces, would cleaning make a difference? So they put Listeria on their paper stickers, and then they treat them with water, soapy water, or a disinfection agent, which would, approximate routine cleansing. The idea would be you'd leave this on a surface, and the cleaning person would be instructed not to avoid it, right? And not to take it off, of course. After cleaning and disinfection, they could get more bacteria detected on the paper than the control, which was just a ceramic tile, right? Which, you can imagine the tiles on the wall of a food processing area. So they spike the tile with bacteria, then they wash it. They don't detect as much bacteria after that. These stickers are about 50 square millimeters in area, so pretty small. And they note this is smaller than the area that's usually suggested for swabbing. When you swab a surface, there's a prescribed area you're supposed to do, right, in multiple directions over a certain amount of time. And so they thought, well, what happens if we pool the stickers? And instead of using larger stickers, we do more small stickers and pool them for DNA extraction. And they have cute hand-drawn pictures in this paper, which are really remind me of my youth as a scientist when we didn't have computers to draw images.
Your undergraduate notebook.
That's right. It's just cute. So
but they're
cute.
so clear.
They're very clear.
You don't need any fancy graphics to make the point.
No, you don't. You don't need any fancy graphics. That's absolutely true.
It's very Khan Academy-like. How
yes, that's right. Khan Academy.
Yeah. Very Khan Academy-like.
Well, the whole method is very straightforward, right?
Oh, yeah.
And this isn't going with it. I think sometimes the simpler is really the most effective. Anyway, they took 6 stickers, they pooled them, and essentially they get the same kind of sensitivity as they do when they keep them individually. So that's good because, it can reduce the number of stickers you have to sample, right? You can imagine having thousands of stickers, and you could pool them and cut the numbers in half. And they also say if you pool stickers, then maybe you detect minor contaminants. Okay, so then they do a real world experiment. They put these stickers in an environment, a real environment, door handles, light switches, especially of toilets. That's a good one, right? Because people come in high touch surfaces where people are not washing their hands as often as they should. And they put these stickers and they left them there for 1 to 7 days. And they looked for Listeria. They first looked using sponge sticks in parallel to the papers. And they looked for PCR, of course. So they can find both species from several locations, both Listeria and E. coli. The stickers detected similar or even higher concentrations than with the swab system. And when they looked from 1 to 7 days, it didn't matter how long the time was. You can still detect it very efficiently. So they have a nice table here showing the stickers are really better than swabbing.
Yeah, and probably faster.
Yeah,
well, they point that out. They say a major advantage of stickers is in handling. They are easy to distribute and collect. No further processing steps such as centrifugation are necessary for subsequent DNA extraction.
Yeah.
And probably better reproducibility than the swabbing.
Now, throughout these studies, they've used a mutant of Listeria. It has a known change, which they could use as a control to make sure that it's what they put there. And they said in this proof of concept study, they could actually look for that because 10 years ago,
they had an oops. An
oligonucleotide was accidentally dropped or distributed. They call it distributed over 10 years ago. It was a 100 base pair oligonucleotide, and they can still find it on the door handle of this room with these stickers.
Wow.
10 years ago.
So those of you who do PCR for a living, pay attention. Your debris could still be floating around.
Oh, it's very persistent. You have to be very careful. Yeah. In fact, people who do PCR have to do it in a separate clean room.
Yeah.
Because otherwise you will get contamination from the lab.
Lots of UVC to bust up the DNA.
Early last year, I had a number of people here at Columbia. Well, we're all screened randomly for COVID and for SARS-CoV-2 infection. And a number of us had positive tests with very high CT values. And all of those people had plasmids in the lab from the viral genome.
Absolutely.
Wow. You can tell who's been working and who wasn't.
Yeah, so I just walked into the lab. I didn't do any lab work, but it's probably floating around, and you inhale it. It's in your nose when you go for the nasal swab. So they said they try and control for that. It's hard because what are you going to do, right? If you're working in a lab, I don't know what else you can do. Anyway, so DNA is everywhere. It's a real problem, as we say over on TWiM. So that's this feasibility study basically shows that plain paper could be stuck on surfaces and used to assay for contaminants. And they note that, of course, this doesn't pick up living cells, but it will detect living cells, dead cells, and viable but not culturable cells, everything. Mm-hmm. And they note that even though in the past this was known as a disadvantage to pick up non-growing cells, but today VBNCs are a threat, right? So,
and especially in the food industry, And this is really important to note is that the European Union is thinking about banning the washing of pre-salad mixes, the washed lettuces. They are outlawing the washing in dilute bleach. And that's currently going through the European Union. I had a conversation last week with and so I said, how are the Europeans going to have pre-made salads? And the answer is they're going to have to wash it themselves.
And what's the rationale, Michael?
Because they're looking at the toxicity of the residual chloride. We don't know if
generating too much waste or the ingestion of the residual.
It's not altogether clear to me. I asked that question of him, and I got both answers. It's residual as well as in contaminating the product with adducts.
It's not a matter of making VBNCs, is it?
No. It's not a matter of VBNCs. It's the issue of the chloride doing the bleach is doing something bad to the food that we.
Michael, ever since you and I were at an ASM meeting years ago, we saw a talk
by the FDA.
He showed a package of pre-washed lettuce. It had been opened up and it had mud in it.
Oh yes.
I will never just eat lettuce out of any bag from then on. I always wash it.
Me either.
I don't trust them.
I think all microbiologists wash their food because we know what's on it and in it.
Yeah.
Unless it's already fermented. Then we'll be happy to take the mixed microbial end products called beer.
The other thing I just want to point out before we move on, they say that used stickers can be used to detect flying DNA, right? As they found in this case, which they say is not likely to occur in the food industry, but where you process these stickers, you're going to have to be careful so that there's not you don't have oligonucleotides from the bacteria you're looking for or plasmids. They could contaminate the paper. And,
jumping genes, horizontal transmission
right.
through the air.
Well, all of these things have to be controlled for, and in good clinical labs, they have a QC program that actually looks for this. And the labs that routinely do novel genetic isolation and novel genetic detection.
Yeah, exactly. All right. So then I found another paper that uses the same approach. This one published in Scientific Reports, a new long-term sampling approach to viruses on surfaces by Sommer, Bulbul, Bromberger, Mester, and Rosmanith. And
from the same group. Same group?
Same group. Yeah. The University of Veterinary Medicine in Vienna, a few other people also from Vienna. But essentially the same group. They just tried it on viruses. A bacteria paper came out first. And they say here, the gold standard, of course, for detecting viruses is the plaque assay. But, they lament. It's expensive. It takes time. Right. It's not great. You can't do it for all viruses, right? Not all viruses form plaques.
And so if you're looking for SARS-CoV-2, you can't do it outside of a biosafety level 3 lab.
That's right. And so people are looking for other methods. Of course, PCR has come into play for looking on surfaces, but as they point out, we're still using swab sampling. And as we have just heard, the swab is not so great. And so maybe paper is better. So they say we tried paper with bacteria and it seems to work really well. So we decided to take a paper-based sticker approach for viruses. And I must say they've gotten more sophisticated with their figures.
Yeah,
They're in color now. They're in color. They seem to have used a computer to draw squares and phages and swabs and so forth, which not as quaint, but that's what happens when you progress. So same idea. You take plain paper stickers, you put them on surfaces. You can leave them there for a long period. You can extract the DNA and do PCR. And so they compared swabs and stickers. And they're using here a bacteriophage, right? So safe, easy to do plaque assays. And they compare paper sticker sampling using plaque assays first. And they measure PFU. And in fact, neither the swabs nor the stickers give you good recovery rates over a broad concentration range. And you need a lot of bacteriophage, 10 to the 8th and 10 to the 7th PFU per 100 microliter give you the most reliable recovery. And that's a lot. That's a lot.
You may not find on surfaces that much. You want to look for less, much less. So they conclude that the the swabbing method and even the paper with plaque assay is not the way to go. So what are you going to do? So, and they talk all about the paper being, you probably have to use the right paper and the recovery. And they say it's a little beyond the scope of this paper, but let's try PCR. Because they say you could probably improve the recovery rate on paper for plaque assays, but let's try PCR, which of course is not going to pick up in just infectious virus, but all pieces of nucleic acids. But that's what they did. And by the way, the detection limit for the bacteriophage is P100 with the plaque assay is 10 to the 4, 10,000 PFU per 100 microliters. So it's a lot. And that's why they say they use PCR. Okay, so then they put phage onto the paper stickers from 10 to the 8th down to 10 to the 2 genome equivalents per 100 microliters. And then they extract the nucleic acid from the paper and do PCR. And they do the same protocol with swabs to compare the two. And they get a good, what they call a coefficient of determination for both the swabs and the paper. It's linear. The detection is linear using PCR for both sampling materials. And the recovery for stickers is 15% and swabs is 18%. So the stickers have comparable sensitivity to swabs. I was a little surprised that that's pretty low, right? 15 and 18% of what you put in. Probably you could improve it, but they don't try and do that. This is just a proof of concept here.
Well, I wondered why they didn't use the most probable number methodology that is routinely used in water analysis to look for tubes. And it's effectively a sampling strategy, if you will, to determine if your potable water is contaminated. And again, it may have just been beyond the scope of this particular study. And it would be in a more comprehensive paper looking at that because, microbiologists have been using MPM-based sampling to assess the safety of everything from hot dogs to the bottled water that comes out of, the Coca-Cola factory.
And in general, how does that method work? Most probable number.
It's a statistical, you inoculate so many tubes with your water sample, and based on the frequency of positives that it has a statistical probability chart that follows a Poisson distribution, because remember, sampling is a stochastic process. You can never sample precisely because the microbes are distributed randomly. And so it doesn't conform to normal statistics time and time again. So it's a Poisson distribution, and it takes into consideration that particular peculiarity that microbes just go everywhere. It's sort of like Vincent was positive with his SARS sample because he had been in proximity to enough nucleic acid and he inhaled it and it was persisting in his nasal cavity. And when they randomly sampled him, he was positive. But when he went back 3 days later, he was no longer positive. And so that's effectively what MPM takes into consideration. Was Vincent a true positive or whether he was a false positive? And the MPN has done that enough to plot limits of confidence and they give you a confidence interval associated with the concentration based on the number of tubes that light up. And that's effectively routinely done for plaque assays for viruses in water, as well as bacteria in water and hot dogs and all sorts of other things.
I see.
By the way, this bacteriophage they're using, P100, is a phage of Listeria. So they used Listeria in the previous paper that we discussed, and they're now using it plus a phage to do these experiments.
Well, they can grow Listeria quite well in their lab, as they showed us previously, and so it just only makes sense that they plaque it on it.
However, they did purchase the phage. They didn't grow it up themselves. Okay, then what about if you clean the surfaces? Just like we did with the bacteria, if you clean the surfaces, and they use ceramic tiles as their experimental surfaces, and they put stickers on the tiles and contaminate them with a known amount of virus, let them dry, and then clean them with different methods that would be used in, food handling environments, hospitals, public places. And swabbing it, you lose material if you swab in between cleaning, as you might guess. But when you do the PCR, you get similar viral load despite the cleaning, which makes you wonder, how good is the cleaning?
Well, it might be lysing like the envelope virus, but
Probably.
now you've just got shards of DNA
sticking to
detect.
They're sticking to the ceramic, and you can't really get that off unless you really scrub really hard. Now I have to say that this is a DNA virus, so if you get the similar effect with an RNA virus, we don't know, but they'd have to do that, of course, in order to find out. And in fact, they do say confirmation will require a range of viruses. But one thing we have found that when we use this sticker, we don't get loss of material that's associated with cleaning. So that's good. And maybe this gives us more information than a snapshot swabbing approach. The only thing, I think it's very clever. The only aspect that is a little puzzling is you're going to put these stickers all over the place. People are going to come by and rip them off, right?
Absolutely.
Unless you tell them, these are for sampling, don't touch them. And then some wise guy is going to put their finger on it. I just don't know.
Well, that's true in an academic research laboratory setting, but maybe in a food preparation
maybe
setting,
they
it
behave.
wouldn't be such a big issue.
Maybe they behave. Yeah.
I was fascinated how the Europeans named their disinfectants. I really like the Micro Z is what they use to disinfect the surface. Micro Z. And it's just an active name, like it's going to get rid of the microbes.
They have another one, Budinat Alcacept.
Yeah, I like that one too. That's what they use to clean. And
anyway, it would be very interesting to put stickers in different areas and then look for SARS-CoV-2, for example.
Oh, yeah.
Because we really don't know. There have not been a lot of sampling. I think when people have used swabs, they don't get much. And maybe that's because the swab is too short a period. And you can imagine, if you have a large surface area, you pick one with this, it's just not the right area. Now, a handle or a switch may be better off, but I like this. This is pretty cool.
Yeah, it really, the only one paper was that New England Journal paper that came out in March of 2020 that evaluated paper, it evaluated stainless steel, it evaluated copper surfaces. And that was a nice study. And they actually plaqued viruses for SARS-CoV-2. But, there was, for many, many months, we were nuking our classrooms with harsh chemicals and ultraviolet light. And then after the pandemic went on for a while, we appreciated that there wasn't much fomite-driven SARS-CoV-2. Right. It appears to be aerosols.
Yeah, they're very little fomite-driven. So it's good. We get rid of the hygiene theater, which... Yes. So spraying the clouds of... whatever it was in China, remember those news images? That just wasn't effective. By the way, Mike, I have to tell you, I went to the hygienist the other day, right? The dentist, and they switched from the last time I went. They used some, Listerine, and now they switched to some iodine-based liquid. It's like 2 mLs they give me and they said they did some research and they found that this was better. Of course, you can't be allergic to iodine. Do you know anything about that? What's the iodine?
There's actually published studies and it's about how many orders of magnitude were reduced on the viruses, and they actually published in the journal of some either infection or CID. It was one of the two journals. I'll dig out the reference and give it to you, and it was actually superior over some of the others that were peroxide based.
I see.
The other solvents had more effective activity against the SARS-CoV-2.
And then, they use the cavitator, right?
Yeah.
Which generates a lot of aerosol. So the dentist apparently bought this machine. It's a big thing on wheels, which they wheel up to. It has a long tube with this plastic thing they put near your face, and then they turn it on, and it sucks the air away from her as she,
Wow.
for all of 10 minutes. I can imagine what he spent on those, right?
I will send you a paper that the Brits have done on aerosol generation in a dental operatory. It's a fascinating fluid dynamics study, and they actually measured it. But the simple take-home is because dentistry uses lots of water to cool the hand pieces, the solution to the infection is dilution. And so you're diluting the infectious dose, but no one's done the study to understand how long or how much of an infectious dose needs to be in the atmosphere in order to infect you. But the Brits, from what I've been reading, are actually going to do the controlled trial where they vaccinate and then infect people.
Okay. Good luck.
I guess it got through their IRBs.
I hope it doesn't. One of the outstanding questions is, as you know, even fully vaccinated people can get infected and get very mild COVID. People who aren't vaccinated and get mild COVID can go on to long COVID. So is that going to happen with
if I was sitting on an IRB, I would say I don't have enough information to determine the risk.
I would agree with that.
Yeah, I sure wouldn't sign up for that trial.
No.
And by the way, when I go, the last 2 times I've been to the dentist, he doesn't show up. He's probably out in his country home. He lets the hygienist do everything. He's probably too scared to show up because he's an old guy, right?
Yeah. Well, rightly so. We know the age demographic is a real risk.
Yeah. All right. That's TWiM 250. You can.
That's a big round number.
It's a cool number. Hey, later today.
Yes.
Daniel Griffin and I are recording
800?
TWiV 800. Yeah.
Oh,
no, really?
Congratulations. Yeah, Daniel got 800. I couldn't fudge it to not give it to him. And I said, you earned it. You've been working hard.
You too have really earned it. Really appreciate
800 TWiVs.
We were in 5-something when the pandemic started. So it shows how often we've been doing them. And, 250 is not bad, but makes a big difference to go once a week, I tell you.
It does
3 times a week,
3 times a week is a lot of paper.
We're not going to do that forever, but until such time as people. So right now there's still a lot of demand. People are still really interested.
SARS-CoV-2 has not gone away.
No,
so
it hasn't.
It will determine how many episodes.
Unfortunately, Charleston is still in the ditch. We have our test positivity rate is still about 18%, where our state is seeing over 4,000 cases a day. And
oh gosh.
And our ICU is completely full, and we're on, they're asking people to be especially diligent, and everybody is very, very cautious.
So these ICUs are mainly non-vaccinated people, I presume?
Unfortunately, yes. Charleston is still only at about 47% fully vaccinated. We haven't tripped 50% yet. And yet we have people running out trying to get ivermectin.
Yeah,
they're literally going to veterinary stores, Fleet and Farm, and they're trying to buy deworming medication for horses. And I said, do you have worms? I mean, you only use ivermectin if you have worms.
The veterinary supplies they can't keep it in stock because people are getting it and they need it for animals, right?
Yeah, they need it to deworm the animals.
And the doses, the veterinary doses are too high for people, so people need to be careful.
Well, they're designed for horses, which are much bigger than people.
They are. They are bigger than us, yeah.
Wow. Well, keep up the great work, Vincent.
Thank you. Microbe.tv/TWiM is where you'll find the show notes. You can send us questions, comments, twim@microbe.tv. And if you enjoy what we do, consider supporting us financially, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thanks, Michele.
My pleasure. Thank you both.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.